Hello guys, welcome to today's video. In this video, I shall be taking you on genetic code. Remember in my previous video, I talked about central dogma of science. In the central dogma of science, we said that DNA is usually replicated from existing DNA. Then DNA can be transcribed or copied into an mRNA, and the mRNA can be translated or read or decoded to give you a functional product like protein. Now, transcription and translation is what we call gene expression. And I said that that takes place in the ribosome. It is in the ribosome that the RNA gets translated or decoded or interpreted into a protein using the guideline we call genetic code. So in this video, I want to take you through a journey on this guideline. Now, let us look at properties of the genetic code. Let's start from there. What are the properties of the genetic code? Properties of the genetic code. First of all, the genetic code is a triplet codon. It is triplet in nature, triplet codon. Meaning that if we have our mRNA, let's say this is our mRNA like this. This is the mRNA that was constructed or built in the process of transcription. If this mRNA has this sequence this way, let us say CUG, let us say AUG, A, C, C, U, G, and probably U, A, G, and so on. Let us say this is the mRNA that left the nucleus after transcription and headed for the ribosome. When it gets to the ribosome, it's going to be translated. That is the essence of what we're talking about, genetic code. In the translation, we said the codon, the message on the mRNA will be read in triplet codon. So one of these base and another base and another base, it will take 3 bases before you have 1 unit of a message. That means you need 3 of these bases as a codon, triplet codon, before you can be able to have 1 unit or 1 building block of protein. And a building block of protein is called an amino acid. There are several amino acids, and then one of them is always built or always coded for by a triplet codon. So there are sets of triplet codons here. Each of them is going to code for a corresponding amino acid. So the first property of the genetic code is that it is a triplet codon. Let us come here. Here is a breakdown on the genetic code of triplet codons. This angle or this wing represents the first nucleotide base on this message that has just come from this our mRNA. Remember, this is our mRNA from the nucleus that has just been imported into the ribosome for translation so that protein could be made, and then the gene that was in the nucleus of the with the DNA could be interpreted or expressed. So the first base is on this wing. This is for the second base. That's for this third base. So if you pick the first base, let us say a U, this is uracil, you come to the second base and you still pick a U. If the first base is a U, the second base is a U, and the third base is a U, that will give you UUU as a triplet codon. This triplet codon will code for the amino acid phenylalanine. Similarly, if you have a first codon to be U and a second codon a first base to be a U, a second base to be a U, and a third base to be, let us say, a C, that is still going to give you UUC, and that translates to phenylalanine. Similarly, if your first nucleotide base is a U and the second nucleotide base is a U and the third one is an A, that will give you UUA as a triplet codon, and that translates into leucine. So that's the way you just go. You have first base, second base, third base. So let's come to this hypothetical mRNA here. Here we have a C and a C and a C, that is first base C, second base C, third base C. So when you put this together, that gives you CCC, and we are told by this genetic code that that will give you proline. All right. So just from that first status, even though that is not supposed to be our first protein, but let us say if we are following this thing serially that way, this place now ought to be proline. However, the genetic code that's not just the only property of the genetic code. It has a start codon. In other words, irrespective of irrespective of the base, the triplet codon that you have, if you are yet to encounter a start codon, there cannot be translation. Translation only starts when you have hit or encountered a start codon. And according to the code we have here, the start codon is always AUG. And AUG codes for only one amino acid, which is methionine. All right. So once the mRNA gets into the ribosome, it is going to be read, and in the process of the reading frame will start from where AUG is encountered. So when we come here, we have here AUG. Before AUG here, we have CCC, which would have given us proline, and we have CUG. Let's check it out. CUG, which CUG would have given us leucine. But you see, ordinarily, we are supposed to have proline here as our this thing. We're supposed to have leucine here, okay, as the next amino acid. And then before we have this methionine here. But because of this rule that there's a start codon, this proline will not exist. This leucine will not exist. The protein will only the building of the protein is going to start only at this methionine. Methionine will always, as a rule of thumb, be the first amino acid in every chain of polypeptide or in every protein. So this is where the reading will start, here, right here with methionine, AUG. So AUG is our start codon. The next one we encounter will be AAA. That is A and A and A. AAA says lysine. Okay, so we are now going to have the next building block. The next amino acid will be lysine. After that, we have ACC as the message that is coming on this mRNA. So ACC, ACC says threonine. All right, so next here we are going to have threonine. So this is ACC, threonine, AAA, lysine, AUG, methionine. Then you have UGU. You have UGU. Let's go check it out. UGU, which says cysteine. So here it will be cysteine. Then it's possible that now the next one we have UAA. Let us fix the next one here to be UAA. Now when we fix that UAA, it says stop. So that becomes our stop codon. Okay, because according to the board, the code here we have UAA, meaning stop codon. It doesn't code for any protein, doesn't code for any amino acid, so it marks the end of your translation. So from this hypothetical mRNA we have here, we are going to have a polypeptide with just 3 or 4 amino acids. We're going to have a polypeptide of 4 amino acids, or a protein of 4 amino acids. The sequence of those 4 amino acids will be methionine, lysine, threonine, and cysteine. That means assuming that this is the 5' end and this is the 3' end. Okay, so remember we hit a stop codon here, UAA, meaning that one of the properties of the genetic code is that it has a stop codon. So triplet codon, start codon, and stop codon. And I just mentioned something here about direction of this mRNA. The direction of translation is always from 5' to 3'. So genetic code has polarity. Okay, so there is polarity. It doesn't just read it or translate in any direction at all. You will agree with me that if we are going to translate from the direction 3' to 5', that will give us a different protein altogether. For example, from 3' to 5', if you go this way, this would have been UGU, this would be GCA, and then A, but this will be GUA, and GUA will give you something different from methionine. All right, so always start translation from 5' to 3'. Polarity is very important when you are dealing with DNA, when you are dealing with chromosome. Remember, in the case of transcription, the new transcript, the new mRNA is built from the direction 5' to 3'. However, the existing DNA there was read in the direction 3' to 5'. Similarly, in replication, new DNA is also being built in a direction 5' to 3', whereas the old DNA is copied from the direction 3' to 5'. So in each case, in all the time, polarity is important. Take note when you are dealing with substances like this. Polarity is very, very important. Now, next property of the code is non-ambiguity, or we can say the genetic code is non-ambiguous. Non-ambiguous means that each of the triplet codon codes only for one amino acid at a time. So one triplet codon cannot code for a particular amino acid and code for another amino acid. Example, UUU here is coding for phenylalanine. You cannot find UUU on this table coding for any other amino acid apart from phenylalanine. Same thing, AUC is coding for isoleucine. AUC codes only for isoleucine. There's no any other place on this table that you're going to find AUC coding for some other amino acid other than isoleucine. So that's the meaning of the non-ambiguity or non-ambiguous nature of the genetic code. It means that each of the triplet codons codes only for particular or specific amino acid at a time. Closely related to this property, and sometimes could be confusing for a lot of students, is what we call the degenerate nature of the genetic code. So the genetic code is non-ambiguous because each triplet codon codes for one amino acid at a time. However, the genetic code is degenerate, meaning that on the opposite, one amino acid can be coded for by multiple or more than one triplet codon. In this case, let's take leucine. Example, leucine is being coded for by CUG, CUA, CUC, CUU, AU, UUG, and UUA. So about 6 triplet codons, different triplet codons, are coding for leucine. So leucine is coded for by 6 different codons. However, not each of these codons cannot code for any other amino acid other than leucine. You know, for clarity's sake, let me give this social example. If you are familiar with the situation in polygamous homes, you know that one man marries multiple wives. So the man is, we can look at the man in this light of degeneracy. So the man is just like an amino acid in this case, and he is married to 5 or 4 or more women. But the women, they are like the triplet codon. Each of the women is only loyal to the man. Let's say our man's name is Mr. X. Mr. X has 5 wives. Each of the wives are loyal to Mr. X at a time. Each woman, each wife marries only one man at a time. In this case, the man happens to be Mr. X. However, Mr. X is degenerate because he has the freedom, according to that culture, to marry more wives. So he is degenerate. He can be he associates with multiple women, but women, just one woman can associate with one man at a time. All right. There are other cultures that the reverse is the opposite. In the polyandrous cultures, where a woman cannot marry multiple men, in which case each of the men remain non-ambiguously committed to that woman, but however the woman is degenerate, relate with them degenerately, she can marry multiple husbands. So that is the meaning of the degenerate property of the genetic code. Another property of the genetic code is that it is non-overlapping. Non-overlapping. Okay. This means that when you are reading this triplet codon, each of the triplet codons stands alone. It doesn't overlap. This means that one of these bases here, let us say I pick one of these nitrogenous bases here, okay. If I pick this cytosine, I'll pick this 1, 2, 3. That makes a triplet codon. So I cannot, for example, pick C, let us say A, U, G. So non-overlapping means these 3 form a codon, these 3 form another codon, and another triplet here form a different codon. There cannot be anything like after these 3 have formed a codon, and then another codon starts from here. One of these base cannot be party to more than one codon. That's the meaning of non-overlapping. You cannot have one base coding for one triplet, being part of one triplet codon and still being part of another triplet codon. If we have something like this, this would be overlapping. You see, in this case, you have CCC as a triplet codon, CCA as a triplet codon, and then CAU as a triplet codon. This is overlapping. Okay, but we said that this is non-overlapping nature of the genetic code. The other one is that it is commaless. Commaless. This means that there is no codon, there is no triplet codon that in the midst of the sequence that doesn't code for any particular amino acid. There's no triplet codon that serves as a free space, that serves as a comma. Okay, all the triplet codon, all the codons, they code for a particular amino acid. So once you have the first codon, it is continuous, automatic, continuous till you hit the stop codon before you stop. So this property says it is commaless. Another property is that it is universal. Okay. Universal entails that research has shown that this genetic code, the way we are looking at it now, is applicable to pretty much all of life on Earth. This code is true from bacteria to man, the lowest form of life to the most advanced form of life. The genetic code holds true. That is the meaning of the universal nature of the code. So don't forget, in translation, you start translation only when you have hit the start codon, which is methionine. And then you continue sequentially without comma until you encounter a stop codon. If you encounter the stop codon, if after the stop codon, there are still codons, there are still triplet codons, they will not be part of the amino acid. They will only be part of the amino acid if downstream, that is after this first stop codon, you encounter another start codon again. If you encounter another start codon, that means you start another fresh translation from there. From that second start codon, you translate until you encounter another second stop codon. In that case, it means that that amino acid, this mRNA, that mRNA coming from the nucleus and heading into the ribosome will produce more than one protein. It will produce 2 proteins or more. Also remember that if you have codons before the start codon, they will be meaningless. They will not make any sense. That's because you can only start codon. The reading frame starts from the start codon, which is methionine. So these are basically what the genetic code is all about. It is a way in which the information is being interpreted in the ribosome in order to give us proteins. Proteins now being essentially the product in which, or the form in which, the gene is being expressed. Okay, like the video and subscribe and share. I'll see you in my next class. Thank you.